Good afternoon, students. Let us continue with histology lessons. Now this is actually a repeat class because in the last time before when we had the practicals, the last things we had done were tubular structures in the neck which came under head and neck, and we had done trachea from the respiratory system and esophagus from the gastrointestinal system. At that time I had already taught you all the layers of the tubular part of the gut, that is the gastrointestinal tract, and I had told you that there is a plan, there is a uniform structure throughout the tube with regional modifications or variations depending on the function of that part of the gut. So today actually I am repeating that. You do not have the luxury of a practical to go with this, but I want you to try to recall because you have seen the slide and you have drawn this diagram already in rough and in fair. So this is a revision to establish the learning, because then from the next class onwards I'll be able to go faster when I deal with more complicated parts of the gut such as stomach onwards. So let us start with histology of the gastrointestinal tract. Now the overview of the alimentary canal is that it has 4 broad layers. As you can see in this particular diagram, which is very small here, there is an inner pinkish layer which we call the mucosa. Then there is a yellowish layer which we call the submucosa. Then a brownish layer which is the muscularis externa, and another thin pink layer on the outermost which is the serosa or the adventitia layer. Now these 4 broad layers are similar throughout the length of the digestive tract, and we will see local specializations as we go. So today we will look at the specializations within the esophagus, but otherwise the esophagus, which starts after the pharynx, if you remember, at the lower border of the pharynx, the esophagus starts, and it will have the same 4 layers up till its end where it joins the stomach at the cardioesophageal junction. Now the lumen of the entire alimentary canal is lined by the epithelium. We know this. The innermost layer called the mucosa essentially is the epithelial lining which comes into contact with the swallowed food. So here inside the lumen of your digestive tract, this epithelium is the first thing which touches the contents of your alimentary tract. Inside will be the food, and this inner part of the circle will be the epithelium. Deep to it is a loose connective tissue which we have always called lamina propria. This is a richly vascularized zone and contains glands as well as lymph vessels and sometimes lymph nodules. It's the lymph nodules which will be visible. These vessels are too thin. They are even thinner than vascular structures like capillaries, so they don't get noticed in a histological section. But glands and lymph nodules will definitely be visible. Now surrounding this coat, that means outside of the pink, there will be a brown, which is a thin layer of muscularis mucosa. A new term we have come across here, because so far we thought muscle of a muscular tube will be on the outside. But here, just supporting the inner epithelium, there is a muscle layer. And in that there are 2 layers. So there is an inner circular layer and an outer longitudinal layer of smooth muscle. So these 3 together are going to be called the mucosa. That means the lining epithelium, its lamina propria and its muscularis mucosa together is equal to 1 layer, the innermost first layer called the mucosa. Then there is a next domain again of connective tissue. This one we call submucosa. It is more noticeable, it is more prominent. It is composed of dense, irregular, fibroelastic connective tissue. This one in the entire gastrointestinal tract does not have any glands except in 2 places. One we will see today in the esophagus, and next we will see it again in duodenum. So you can remember it when we come to that part. Now this fibroelastic connective tissue layer means that it is providing some mechanical support to the structures inside of it, which is the mucosal layer. The mucosal layer itself has very minimal connective tissue in the lamina propria. So the submucosa's connective tissue, which is consisting of fibers including collagen fiber and elastin fiber and reticulin fibers, that's why it's called fibroelastic. So this is the main substance, the matter in the submucosal layer, but it will also have blood and lymph vessels, because this is the layer which brings in the supplies to keep the epithelium alive. We've always said that generically Anywhere there is epithelial tissue, it is cell rich. The intercellular spaces almost don't exist. It has tight cellular junctions, and it does not therefore allow or have a place for blood vessels or lymphatics or nerves to go in between. Nerve fiber endings still manage to get in between the epithelial cells, but vasculature comes and stops in the lamina propria region. So the submucosa is where branches of the incoming vessels, arteries and veins will become smaller and smaller, and they will ramify here, and from here their finest branches will become capillaries, which will go into the lamina propria and supply the epithelium or layers of it. Now in the esophagus there is a certain kind of epithelium. I think if you already remember, you will remember it is the stratified squamous non-keratinized variety. But no matter what kind of epithelium it is, and in some regions of the gut, it is quite an elaborate, large volume containing epithelium, because it has both the functions of secretion as well as absorption. So when it has so much of epithelial cells and so much of modifications and specializations, it requires a very rich blood supply, especially when there is secretion happening. So all the blood supply is coming in through the submucosal layer. The blood and lymph vessels are therefore important. It may not be very prominent. In this small picture, I don't know if you can make out, please try to see. There are arteries and veins together in this yellowish orange layer where you see these vessels. So you're seeing cut sections of the smallest branches of the incoming arterioles and venules, and in the slide also you will be able to see them quite alright. You will be able to recognize vascular tissue within the submucosa. Plus, now the inner epithelial layer has some functions of secretion, which will require a autonomous nerve supply for glands to secrete. So we have a Meissner's submucosal nerve plexus. So in the second layer, which we call submucosa, we have a nerve plexus which has a name. Since it is in the submucosa, we call it submucosal nerve plexus. If you call it that, that is enough. But if you remember Meissner, that is good, because he is the scientist who named it, who discovered it, and therefore it is named after him. This is a very fine network of nerve fibers available to serve all the functions from submucosa inwards towards the mucosa. Outside of this connective tissue domain called submucosa, there is the thick muscular layer, which is very easily visible even to the naked eye, which is the most prominent part of the gut tube. If you just look at the gut tube, the muscle is the most prominent part. If you look at just the esophagus with the naked eye in your dissection, it will just look like a muscle tube. All these layers will not be apparent. It will just look like muscle. On the inside, there will be a thin membranous epithelium, which doesn't look very significant when you look at it with the naked eye, except that it will throw some folds of mucosa on the inner lining. It's not shown in this picture very prominently, but there are folds of mucosa which help to increase its surface area, and which is the position it has in a collapsed state. When there is nothing filling the gut, the mucosa internal lining will be thrown up into folds. But when food is passing down it, either immediately in the esophagus, which is almost sometimes semi-dry food, but the chewed bolus which has come down from the oral cavity, passed from the pharynx into the esophagus, that when it distends the muscular tube, then these folds disappear in the mucosa. Whereas the outer muscular layer is a much more prominent layer, visible to the naked eye and consisting again of two layers. So we call it the muscularis externa because it is outside and it is not the interna. There is no word like muscularis interna. The one we use is muscularis mucosa. So you have to remember the terminology. The muscle for the inside, which is an intrinsic part of the mucosal layer, is called muscularis mucosa. Name is self-evident. Whereas the main bulk of the tissue holding up the shape of the gut is the muscularis externa. And this thick muscular layer is where peristalsis happens, which you will learn in physiology alongside. So the peristaltic activity is contributed by this thick muscularis externa. This squeezing, and in a longitudinal wave fashion and in the concentric or radial fashion, moves the contents of the lumen along the tract. Now the two layers of this are an inner circular and an outer longitudinal layer of thick muscularis externa. The outermost layer in this diagram also, it's a thin one. It is a thin connective tissue layer which may or may not be surrounded by a simple squamous epithelium, which is called the visceral peritoneum. Now, we are doing this alongside with your thorax gross anatomy. So, in the thorax, the esophagus does not require a peritoneum or a peri anything. It is only having a simple adventitia, a simple serous layer, serosa or adventitia. But in many parts of the intra-abdominal gut, in fact the majority of it, the tube is hanging loose and free within a liquid containing cavity called the peritoneal cavity. So the tube lined outside by a layer of thin simple squamous epithelium which is secreting outward into the cavity, and the abdominal cavity lined inward by a second layer of the same series. So between the two secreting layers is the fluid, and the inner layer is the outermost covering of the gut tube. So this, I think you're already studying it probably. Maybe you'll come to it later in embryology, when you will see how the gut tube forms a suspended tube within a body cavity or coelomic cavity, which will in the abdomen become the peritoneal cavity. So as far as thorax is concerned, you don't have to worry about it. The part of the gut you see in the thorax is only the esophagus, and here it does not have a outer peritoneal or any secreting lining. We just call it an adventitia. This consists of simple connective tissue, and okay, on this slide we have not described it, but we'll describe it when we look at esophagus in detail. A connective tissue with fibers which is holding the whole shape of the esophagus together, and into which it will get pierced by the incoming vessels, nerves and lymphatics. So the tube has to be penetrated by the supplies to keep all these four layers alive and healthy. So if you have got a sense now of the four broad layers which constitute any part of the digestive tract, we'll move on to the specifics. Like I said, in the innermost layer, which we call the mucosa, we should be having a lining which is looking wavy here. Here in the intestine it will be in the form of villi. In the esophagus it will be in the form of folds. In some other places there may be a slightly different architecture, but the point is that it will be a lining of epithelium. And since we know that all glands are extensions of epithelium, the glands which empty into the lumen will be occupying the lamina propria, but their excretory ducts will be going out and exiting from the epithelial lining, the internal luminal lining. So we have the epithelial layer, the lamina propria, and outside that we should have a muscularis mucosa layer, this one, right? These two layers. In this also there is an inner circular and an outer longitudinal arrangement. Now this varies in different parts of the gut. It is not very prominent in many parts of the gut, but it exists. Whether minimum or maximum, it exists, and it is the third point in the innermost mucosal lining. Now in the lamina propria you can see aggregations of lymphoid tissue, which we call a lymphoid nodule, but you'll also see a lot of scattered singular lymphocytes and other types of WBCs which we cannot identify at low magnification or at HE staining. We just get to see a lot of random scattered cells. All this is part of the gut associated lymphoid tissue, the GALT, which we have mentioned earlier when we talked about general tissues and we talked about lymphoid tissues. There were aggregations and organs made of lymphoid tissue, and then there was different nodules interspersed, placed in certain areas, especially close to the luminal surfaces of some tubes. So in the respiratory tract there were some lymphoid nodules. We called that BALT, right? Bronchus associated lymphoid tissue. In the last class I had mentioned it. So that you leave there. Now you focus on the fact that there is a whole bunch of gut associated lymphoid tissue which is seen from esophagus onwards. In the slide we'll try to locate it for you when we show you in the practicals, but in the picture when you draw a standardized classic picture of the microstructure of esophagus, try to include one small lymphoid nodule to show that you know that this is the layer which contains lymphoid nodules even in the esophagus. So muscularis mucosa, then mucosa. Mucosa is the second layer which is coming out. In that again you will have some glands. These are not the same as these glands. The mucosal glands are within the lamina propria of the mucosa. These are submucosal glands. They are also not present throughout the gut. They are there in some portions. We will especially mention them in those portions because they will become identifying factors over there. But remember that this is because it is a connective tissue domain. It will have the space and the looseness to give space for glands to develop. So glands will be here. But again, since glands do not serve muscle layers, their duct will also have to pierce through and empty out here for it to have an effect, right? Mucus secreting glands, what will they do? They secrete the protein mucin, which is a lubricating and smoothing agent. So it will help the food which is swallowed in the esophagus to be coated with a lubricant which will allow swallowing. Deglutition, it will facilitate deglutition, right? Especially if the food is not completely chewed and liquefied in the mouth. This is important for mechanical facilitation of swallowing. Apart from that, mucin is an inert protein, so it will also neutralize any chemical extreme things in the whatever is swallowed in the esophagus. So too acidic, too alkaline, too much of any chemical which is probably harmful, deleterious, the coating of mucin will safeguard the layers of the gut from the effect of whatever is inside the lumen. Now, the muscularis externa. This is the outer two muscle layers, inner circular, outer longitudinal. In between the two, there will be a myenteric plexus. In the previous slide, I had mentioned the Meissner's plexus, which was in the submucous area. This picture doesn't have any nerves showing, but it is showing you the vessels, which I have already mentioned. In between the two layers of the muscularis externa, there will be a myo in this myo is the root for muscle, enteron is the root for gut, digestive tract. So the muscle of the gut plexus. So the muscularis externa has another plexus which is also more prominent in a small strip of area between the two layers, which we will see in the slide. And in that you have the parasympathetic and sympathetic innervation coming in to serve all the muscles of the gut, both the external and the internal layer, plus to serve the glands, which are therefore secretion of various content, various things which will be added to the food. Outside of that is now the serosa. The serosa, we said, is a thin connective tissue domain, and it may be covered outermost by a simple layer or a thin layer of single layer of simple squamous epithelium, not in the esophagus, but generally somewhere it can be covered. Now in this we can make out that there are blood vessels which are running along to the length of this gut piece, for some time, and they are the ones which are therefore visible or present or noticeable inside the serosa layer or the adventitia layer. This adventitia layer, it's called serosa here only if it has a secreting membrane of simple squamous epithelium, otherwise just call it adventitia. This adventitia will bring in the bigger branches of vessels. By the time they penetrate, they will give some branches into the muscle. They will become smaller branches. Here they will be smaller in cut section. So when you see a TS or an LS, there will be smaller caliber vessels. By the time they divide some more and become smaller branches, by the time they come to lamina propria, you will see very, very fine cut sections and capillaries. That's the way the gradation will happen when these supplies penetrate into the thickness of the wall of the gut tube. So, since you haven't seen the esophagus yet, you have seen a bit of it in the neck, but you haven't seen it in the thorax. It's a continuation of this same tube. This we saw. This is a posterior view of it. While it is in the neck region, because this is the floor of the mouth and this is the throat, and then we are going downwards. So when we have crossed the pharynx, we see they cut a window, and to show you from outside, if you cut a window in the longitudinal muscle, you'll see the circular muscle inside of it. So if you're looking at the esophagus from outside to inside, there would have been an adventitia. In this particular picture, it has been peeled off. Then there would be a muscularis externa longitudinal muscle, visible to the naked eye as longitudinal striae of muscle. Inside of that, there would be a layer of circular muscle, that it is, the orientation is subject to the lumen. To the lumen, it is circular. To the lumen, it is longitudinal. And inside of that, suppose we were to cut a hole Inside of this, we would expect to see submucosa, and inside of that, the mucosa. So this is just to give you an idea of how the tube will be. It will be a simple, almost midline, long tube lying almost exactly on the center of the vertebral column of the thorax, just extending from the neck region to the stomach region. It's only a conduit, it's only a passageway for the food to go. It doesn't contribute anything to the food apart from the mucin, which coats the bolus. Now let's look at the microstructure. So this is of course a painting, it's a diagram. Slides don't look so beautiful and so sharp, but you need to label it from all the labeling which is visible here. So if you start from the inside, we have the mucosa, stratified squamous epithelium. I think you already remember this, I've already mentioned it. So we will see it is non-keratinized. So the uppermost layer is a simple squamous line, but it is alive. There are nuclei right up to the surface. There are no dead cells here. This surface will be in touch with your food which you swallow into the esophagus, and it will be coated by the mucin which is being provided by the secretory glands. Now we have seen that glands associated with stratified squamous epithelium can't be within the epithelium, right? It doesn't allow there's no space for any glands, but glands can be there in the lamina propria, from which ducts will arise and they will pierce the layers and they will open to the outside. So at any one given point in the slide, you may not see the openings of the ducts. If you see the stratified squamous epithelium of skin, particularly thin skin, you can see the opening of the hair follicle duct, opening of the sweat gland duct in many places. This is not like that. This stratified squamous epithelium firstly has fewer openings, and you may not see them all in the same field, but if you do, you must remember that they are the ducts of glands which are in the deeper lamina propria. Up to here is lamina propria. I don't see any glands here. We have blood vessels, we have a lymphatic nodule, and we have next the muscularis mucosae. So the this thin line of dots is all the WBCs scattered in the lamina propria. The aggregation of these WBCs, mainly lymphocytes, is the lymphoid or lymphatic nodule. Apart from this, you are seeing cut sections of ducts and cut sections of blood vessels. And how will you differentiate the two? Blood vessels here will be capillaries, so they will have a simple squamous internal lining. There's no tunica media or externa at all. It's only a tunica intima of simple squamous epithelium, 2 or 3 dashes as nuclei. That's all. There's no thickness. There's no material in the wall. Whereas the ducts are simple cuboidal epithelium. Like we've seen, ducts everywhere, almost everywhere. The smaller ducts are simple cuboidal epithelium. That's how you differentiate the two. And the ducts would be of glands which are situated more deeply, but which are transiting through the layers to open onto the luminal surface. So the points for mucosa are stratified squamous epithelium, lamina propria, and muscularis mucosa. Now in this muscle, I hope you can notice, look at your diagrams carefully. It is a TS. It's a TS of muscle. Can you see that they are discrete cut sections, and you can see nuclei in the center of the muscle cell. So if you have a central nucleus, it must be smooth muscle. So we can expect that muscularis mucosa is smooth muscle and therefore involuntary, and it seems to be, at least in this diagram, having only an inner circular layer. But you must remember that actually in architecture, this layer throughout the gut has an inner circular and an outer longitudinal layer, not very prominent in most places. So this one seems to get a little more prominent at the upper end and the lower end of the esophagus. So in the muscularis, there is nothing much to notice, though it is supposed to be served by nerves. You won't actually see it at this magnification. So low, and it's even less than 100x magnification, so you won't see it. At 100x, we will take a small piece. I'll show you in the next few slides. Just a small piece of it, where we will try to see at least 2 or 3 of the layers, because that is sufficient for you to identify this slide when it is kept as a spotter. But this is a kind more of a panoramic or a global view where all 4 layers are seen in the same section. So we're looking at mucosa and 3 bits of it, all the 3 bits of it. As you can see, The submucosa is going into the esophageal fold and coming out again, which means the surface is mucosa and the core of the mucous fold is inhabited by a projection or a continuation of the submucosal structures. So in submucosa, what we get to see is some glands. So in the esophagus proper, please remember the microstructure of esophagus, there should be some glands. These are mucus secreting glands. They don't take up much stain. You remember serous glands take up a good purplish kind of stain, like we saw in pancreas, or we didn't see pancreas yet, or we saw in parotid, right? So that is not the look of these glands. These glands look like the submucous salivary glands, very mucus secreting. Almost all are mucus secreting acini in groups, small basal flattened nuclei because they are wedge shaped cells. The mucin doesn't take any stain, so the center of the acinus looks completely empty. So mucous acini of esophageal glands is one of the features inside the submucosa which will help you identify this part of the gut. Alright, so in submucosa we are seeing mucous acini and we are seeing a lot of loosely arranged irregular connective tissue. This looks like a bunch of adipocytes, little bit of fatty tissue, right? Single adipocytes with single flattened nuclei. Then probably a venule, and this is probably an arteriole because it has a little bit more muscle in it. And adipose tissue labeled over here. Vein and artery, that's the wrong word, it should be venule and arteriole because vein and artery are much bigger vasculature. And excretory duct of esophageal glands. So this duct is cut a little bit here, cut a little bit here, cut a little bit here, but should empty out into the lumen, right? We don't get to see the continuity, but you must remember wherever you see cut sections of ducts, they are leading out from these glands. One feature is the connective tissue of the lamina propria. This whole lamina propria connective tissue is bulging into or protruding into the folds of the mucosa. Like that, the connective tissue in the lamina propria is bulging into or protruding into, like in the form of what we call papillae, into the basement membrane of the stratified squamous epithelium. This helps to hold that whole layer together, no matter how much squeezing of food is happening and how much the layers of the intestine are moving against each other. The stratified squamous epithelium stays firmly adherent to the submucosa, it doesn't get separated because of these papillae of connective tissue. In skin and all, we call them pegs, rete pegs. Don't call them that here. They're simply called connective tissue papillae. Alright, but remember that you have to draw it like that. The basement membrane is not smooth, it is thrown into folds. The surface is smooth, but thrown into broad folds which are visible to the naked eye when you look at the inside of a opened esophagus. So we got mucosa and we got submucosa and all the features of it. Now this was supposed to contain a Meissner's plexus, submucous plexus. We don't get to see much in terms of nerves because nerves don't stain so well with HE staining. But if you did a differential nerve stain, you would see networks of little bit of nerves plexing in this area. Now let's come to muscularis externa. There is a inner circular layer. We are calling it circular because it is circular to the lumen. It is forming the same circle as the lumen. But in terms of tissue, it is an LS of smooth muscle tissue, right? This also looks like smooth muscle to me because the nuclei seem to be inside of the muscle myocyte. And this outer layer seems to be, it is TS. It is TS of muscle, and again the nuclei, are they looking, this one looks a little peripheral. What do you think? The nucleus is peripheral or central? This is looking peripheral, right? Then this one also is supposed to be peripheral. Alright, if you can identify the nuclei to be peripheral to the fiber or to the bundle, then it must be skeletal muscle. So the identifying feature for the muscularis externa layer of esophagus may be that its muscle is skeletal because the rest of it is smooth. But this is not true of the entire esophagus. What you must remember is the upper 1/3 of the esophagus has skeletal muscle. We shouldn't call it skeletal, voluntary or striated muscle in its composition of muscularis externa because swallowing is a voluntary activity. So it is supplied with a nerve supply which is of a voluntary nature, and its action is semi-voluntary. After the 1/3 is crossed, from middle 1/3 of esophagus onwards, it becomes involuntary. Peristalsis takes over. That is an autonomic function. So the nerve supply also changes and continues into the peristalsis of the gut, which is no longer under your control. So the structure serves a function. The upper 1/3 only shows skeletal muscle structure. The middle is mixed. There is skeletal and smooth muscle, and the lowest 1/3 is purely smooth muscle with autonomic kind of nerve supply. Please remember this, very important. In the Viva they will ask you whether it's gross or it is microscopic esophagus. They will ask you this in the Viva. And in between these two layers, I think they're trying to show you some sort of a nerve plexus over here between the inner circular and outer longitudinal. You may be able to see small poorly stained areas which would be the myenteric plexus of Auerbach. Auerbach. We'll come to that in the next slide. So we have a inner circular and outer longitudinal muscle layer. In this particular picture, since we are seeing skeletal muscle, this must be the upper 1/3 of the esophagus. Then outside we have the adventitia, where you have adipocytes, you have some nerve fibers here, better visible because they're bigger branches of the nerves. Their finer branches are here, the finest branches are here, and here you won't see any nerves at all. And you will see bigger cut sections of vessels, more amount of tunica media, which are the again venule and arteriole. But you can call it vein and artery here because the branches of esophageal arteries are the ones which are penetrating the serosa to come and keep on branching till they reach the innermost surface. Some adipose tissues, some loose connective tissue, vessels and nerves in the adventitia. So this is the structure of the 4 layers of the esophagus, the first part of the GIT which we are studying today. This is what an actual slide looks like. So the first thing which jumps out at you is that stratified squamous epithelium E, right? Then you see this papillae, so the very clearly seen, well demarcated basement membrane from the lamina propria, Then you see a submucosa which is looking like a slightly brownish domain, which means the muscularis mucosa is not very prominent. Can't make out. It could be this layer, but not very sure, right? Because if all of this is lamina propria, muscularis should be innermost, right? It should be somewhere here, just inside of the submucosa. In the submucosa, we can't make out clearly much, but we have the inner circular and the outer longitudinal muscle. Now in this slide, he says there is a well-developed rete apparatus with the underlying lamina propria. So maybe it is all right to still call it a rete apparatus, and some books don't use the word rete except for skin. You can, if you just call it connective tissue papillae, that should be sufficient. The submucosa surrounded by the thick external mucosa, muscle, and which is composed of inner circular and outer longitudinal. So from here to here is muscle. If you take thickness from here to here, you see 3/4 is muscle, little bit of submucosa and little bit of mucosa, right? So that is the relative proportion of the 4 layers, but you need to look at it under very low power to be able to make out this. What you will normally be able to see is some of the layers. Now in this we said mucosa has lining epithelium, lamina propria, muscularis mucosa. So that is the demarcation between mucosa and submucosa. It is pretty vague. If you can notice muscularis mucosa, which we need high magnification for, we'll need at least 400x to be able to see it like this, then this would be the area where the lining epithelium, lamina propria, muscularis mucosa ending, and therefore submucosa beginning. There's no special demarcation between the 2 connective tissue domains. And then you have the ducts of the glands, you have the glands themselves, the mucin secreting esophageal glands, you have vessels, you have nerves, all within this connective tissue domain. Then you may have some adipocytes also. The submucosa will have this connective tissue. Then you will see the next layer. Again, the demarcation is somewhat better because muscle stains pink, and you are seeing it in bundles. So muscularis externa is easy to make out where it starts, where it is separated from the submucosa. Now what I wanted to show you in this slide is here the muscle seems to be cut in TS. It is supposed to be inner circular, right? And that is the plan for the entire gut. So we can't break the rule here. So why are we seeing TS of muscle? It looks as if it is oriented longitudinally. But you must remember that the TS of inner circular muscle will happen if this piece is cut in the long axis of the esophagus. Instead of taking a slice like this, I took a slice like this, and this surface of it, I taken a bite and put under the microscope. So then this kind of muscle is cut in TS, and that's what you will see in the inner layer. So the inner circular muscle is always inner circular muscle. Don't label it any other way. You need not call it TS or LS, it doesn't matter. But always label it inner circular muscle, no matter which section it looks like under the microscope. So this is again a little wider view. Same things. Mucosa 3 layers, mucus acini, lymphatic nodule, blood vessels in the lamina propria, connective tissue papillae, ducts and arteries. Only the mucosa 3 layers. In this we are only seeing muscularis in TS, we are not seeing it in LS. You must remember this one will be supplied by that submucosal nerve plexus of Meissner. Now we look at the mucosa and submucosa so that you can see all the labeling a little better. It's not a very sharp picture, but I think the labeling is a little better for you to be able to identify all the parts. So the mucosa, extra points, little bit extra, in case you get it as a theory question, you have to write this. So I already told you the longitudinal folds of the mucosa will disappear when the tube is distended. So these folds which you see, these up and down folds, when the esophagus is open, they are laid flat. So you'll see a continuity, just a slight wavy, not even wavy, some almost smooth internal lining of stratified squamous non-keratinized epithelium. In that, there may sometimes be melanocytes and endocrine cells, but in H&E staining you may not be able to make it out. You can write it as an extra point. The papilla I have already talked about, which are also called rete. Mucus glands present at the upper and lower ends of the esophagus. So if your section is at the upper end, you will see more mucus glands already in the lamina propria also. In our slides, we are not seeing the glands in the LP. We are seeing glands in the submucosa. But if you take a section at the uppermost end and the lowermost end of the esophagus, more mucin glands are provided. They are there in LP, they are there in submucosa also. That is one feature. And muscularis mucosa is thickest at the lower end. This muscularis mucosa is thickest at the cardioesophageal end and has many longitudinal muscle fibers and a few circular fibers. The submucosa, compound tubulo-alveolar mucus glands, that's how you must describe them. Already told you, aggregations of lymphoid tissue, not there in this picture. Plasma cells and macrophages, these are the other WBCs which you will see in the stroma, and all the neurovascular supply. So good number of cut sections of blood vessels and of ducts. This is called the submucous nerve plexus of Meissner. Muscularis externa will look like inner circular and outer longitudinal. Connective tissue domain between the two will contain nerve plexus of Auerbach, myenteric nerve plexus. Right? Upper 1/3 striated, middle 1/3 mix of smooth and striated, lowest 1/3 smooth autonomic muscle fibers supplied by this myenteric plexus of Auerbach. Adventitia, the outermost domain, actually this muscle part is still muscularis externa. So adventitia is only from here to here. It's a very thin layer comparatively. Contains nerve fibers, some adipose tissues and larger size vessels. The vessels you draw here should be of a larger caliber because they are the entering vessels which are going to divide into branches. So we got all of that. This is the comprehensive view you need to draw if you are getting it as a theory question so that you can cover all the points. Mucosa, mucus acini in the submucosa and muscularis externa. These are the 3 points which will help you identify. The mucosa will have stratified squamous epithelium. The only other part of the gut which will have stratified squamous epithelium is the end, the anus, which we don't have a slide of. So there is nothing to confuse with. If you see the slide and you realize that it has 4 layers first of all and you know it is gut, Then if you look at epithelium, it's enough, you know it is esophagus. It can't be anything else. There is no other part of the gut which has stratified squamous epithelial lining of the lumen. Second, to confirm, you look for glands in the submucosa. Yes, they are there. You look for the two layers of muscularis. There is inner circular, outer longitudinal. This is gut. It must be esophagus. The only other place where you have submucosal glands is duodenum. But that one's surface epithelium is completely different, so you can't confuse the two. If you remember these 3 points, you should be okay with microstructure of the esophagus. I hope this was more or less understandable. We have a little time, so we'll talk a little bit about the clinical correlates. There is a condition called achalasia cardia. Cardia means the cardiac end of the stomach. It is not referring to the heart. So the cardiac end of the stomach is the first part of the stomach immediately after the esophagus. So when the esophageal tube becomes the stomach sac, the first bit of it, which is close to the heart, which is why it's called the cardiac end, that part, lowest part of esophagus, undergoes spasm sometimes, which is a neuromuscular dysfunction. This so-called sphincter, which is governing or controlling the amount of food which is released into the stomach little by little, that is a physiological sphincter. There is no anatomical extra layer of circular muscle which will act like a sphincter there. All parts of the esophagus have the same thickness of inner circular layer of muscularis externa. So this cardiac sphincter is not an anatomical feature, but it is definitely a physiological feature. The nerve supply is such that it functions like a sphincter, preventing reflux of stomach contents up back into the esophagus. Extremely important. If there is an achalasia and this functioning mechanism is disturbed, it won't relax during swallowing, and food will pile up in the esophagus and distend the lower end of the esophagus. To the patient, this is progressive dysphagia, inability to swallow, discomfort during swallowing, getting worse and worse over a period of time. And to you, when you take a scan, when you look at an X-ray or a scan picture, you will see that the lower end of esophagus is dilated, which shows you that there is cardiospasm holding up the material which has been swallowed. So this is one condition where it will show up, or it will result from a disturbance in the neuromuscular control of the lower end of the esophagus. It doesn't have much of a histological feature, but you must know that such a condition exists. Barrett's esophagus, on the other hand, will show you a histological change. So if you take an esophageal biopsy, it will actually show you a change which explains what is happening over there. The stratified squamous epithelium of this lower esophagus is replaced by columnar epithelium. The next thing we will do next week is lining of the stomach, and that has a columnar epithelium. In fact, majority of the gut is columnar epithelium. Esophagus and anus are the exceptions to the rule. So this columnar epithelium of the stomach seems to have crept up into the lower 1/3 of the esophagus. That means what should have been stratified squamous has not been laid down in layers, but has been laid down in a single layer as a columnar. This is supposed to be caused by factors which contribute to a condition called gastroesophageal reflux disease, GERD, and that's why the histology also responds and the epithelium also changes. This is supposed to be common in older patients, in senior citizens, and this Barrett's esophagus is the pathological phenomena which leads to the symptoms of the disease. And of course you will learn in medicine later how to deal with it, but how to recognize it is that its histology has changed. Its inner lining has changed to look somewhat like a stomach lining, and therefore it's not going to be able to do the function of the lower end of the esophagus, and that is what will become the symptom of the patient. So what we learn in histology correlates with what can go wrong, and how will it look, how will you figure out, how will you know, how will you ascertain that what I'm thinking is wrong is what is actually wrong, right? How do you confirm your provisional diagnosis of what's happening? So one thing which assists you is a biopsy or a histological study of the tissue, which will tell you that this one's histology has changed, which is why that function is disturbed and giving those particular symptoms. But a biopsy is an invasive procedure. We don't go around picking up pieces of people's tissues. As far as possible, we try to diagnose with non-invasive methods, but still you must know the correlation with what is happening with the cells and with the tissues which is making this patient sick, or other way around, what is happening with maybe the patient's behavior or the patient's environment or what the patient has consumed, which is leading to an insult or injury on the tissues, which is making the tissues respond and react and therefore change. Both ways you will learn these correlations next year in pathology. For today, if you've got the clarity on the 4 layers of the gut and the specificity of points for the 4 layers of the esophagus, which will help you describe it, get marks in your answers, and discuss it confidently in a viva and draw a well-labeled neat diagram with all the features, then I'm happy. Alright, we'll close the class now. We have 10 minutes more. Please put in any questions, queries, doubts you have into your chat, and I'll get somebody to share it with me so I can answer it. I didn't get any feedback for the last class on lung. So any doubts on lung tissue also, both lung and esophagus, share back to me. Either you can message or mail to your website, or probably just put it in the chat associated with this particular class. Thank you. Have a good afternoon, and I'll see you again next week.